Good evening, everybody. We're going to get started here in just a minute. I wanted to introduce myself. My name is Nancy Lomibau. I'm the program director over at the Cancer Support Community in Redondo Beach. That's over on the Redondo Beach Pier. And we offer over 200 free services a month, ranging from weekly support groups to different mind-body activities, as well as workshops, educational programs. So tonight I brought along some of our calendars, which are in the back. But I'm here on behalf of the South Bay Survivorship Consortium, which is putting on this program tonight. We're a group of different organizations, including Torrance Memorial Medical Center, the Torrance Memorial Physicians Network Cancer Care Associates, Healthcare Partners, California Hematology Oncology Medical Group, Cancer Support Community Redondo Beach, the American Cancer Society, Beach Cities Health District, Kaiser Permanente, South Bay, and Harbor UCLA. So as you can see, we bring a lot of people together to the table to bring these presentations to you with the focus on survivorship. We began in 2009, and we started to provide the survivorship education in the South Bay community in 2010. So today we're really encouraging you to visit right outside this room. We want to thank Lilly Oncology, who have brought an art exhibit called Oncology on Canvas. So please check that out after the program. There's also a really neat interactive activity out there. There's a butterfly that you can all help paint in, and hopefully we can get that completed tonight and have a beautiful finished product by the end of the evening. The other thing that's in the back of the room are tables from various organizations. So please go back and visit Torrance Memorial, Cancer Support, American Cancer Society, Harbor UCLA, and of course our sponsor tonight, Council, who provided all of this delicious food for us tonight. They have a table as well back there, so please stop by and look at those materials. So tonight our program is titled, What's in Your Genes? Hereditary Issues for Cancer Survivors. And we have some goals tonight. We hope that you're going to understand about assessing your genetic risk, learn about genetic testing, and learn about the management of inherited cancers. And this topic, it's so important. We have Raluca Kurz tonight. She's a very experienced and innovative genetics counselor that's here at Torrance Memorial. And she's going to be talking about how the knowledge of cancer genetics is rapidly improving our understanding of cancer biology. It's helping us to identify at-risk individuals. It's furthering the ability to characterize malignancies, establishing treatment tailored to the molecular fingerprints of the disease, and leading to the development of new therapeutic modalities. So exciting stuff that she's going to be talking about tonight. And we're also able to use the information about genetics to shape cancer treatments now and in the future. So that's a topic for another program, but she'll touch on that. And the other thing that I wanted to let you know about and mark your calendars is our next program is coming up in August, on Wednesday, August 9th. It's going to be here in the same room. The topic's going to be on nutrition and cancer. And our speaker is going to be Colleen Doyle. She's the Managing Director of Nutrition and Physical Activity for the National American Cancer Society. So we're thrilled to have her coming. And then I want to tell you a little bit about what's at your seats. You have some question and answer cards, those index cards there. We'd like you to ask one question on each card, and if you need additional cards, we can get those for you. But we're going to have a halfway mark in the presentation where we're going to go through and collect some of the questions. And then we'll also have time at the end to collect additional questions. And then all the questions will be answered at the end of Raluca's presentation. but collecting them at the halfway point will allow for us to go through them and sort through any duplicates and hopefully get everybody's questions answered. The other thing that's at the table, we have handouts that are related to today's programs. One of the very most important thing is our evaluation form, because we use that to select the topics for the next program, and it helps give us information about how well received this program is. And the other thing is, if you'd like to be on the contact list for future programs, make sure you include your email or your mailing address as legibly as possible on that evaluation form. And we'll have people who will be collecting those at the end. Please know, too, that tonight the program's being recorded, along with all of our other previous programs, and they are on the Torrance Memorial website. So within a few weeks, this program should be up on the website. A little bit of housekeeping. The bathroom locations, they're out this back door here and to the right. And then to please take out your cell phones and silence them or turn them off. Just a few last thank yous to Lilly Oncology once again for their really terrific art display they brought tonight, an opportunity to be interactive, and those refreshments that are brought by council. And of course, we always want to thank Miriam Slavin, who does such a wonderful job bringing this presentation to all of us and furthering our knowledge. So now, without further ado, I'm going to introduce our speaker tonight. Her name is Raluca Kurz. She began her career in cancer genetics over 20 years ago when the BRCA genes were just discovered. She has practiced as a clinician at both academic and community institutions, and she set up genetic clinics and delivered genetic services to diverse patient populations, both in the USA and internationally. So, an early adopter of genetics as a tool to mitigate increased risk due to hereditary conditions, she believes that education is key in improving access to and delivery of personalized medicine, and has actively been involved in genetic education for nurses, physicians, and other healthcare providers, as well as in the public. So early on, Ms. Kurz noted the unmet need to improve access to genetic testing, both from the clinical and cost standpoint. She joined Invitae in 2012, a novel genetic information company in San Francisco, whose aim is to improve access to genetics globally. She was instrumental in creating and commercializing their cancer genetic panels, crafted with particular attention to the needs of the clinician as well as the patient at time of cancer diagnosis, and to timely integrate genetics into the treatment plan. In 2016, Ms. Kurz began a doctoral program in health policy and management at UCLA's School of Public Health, with a focus on alternate modes for delivering genetic services to augment clinical-based clinical-based care and disseminating genomic medicine across patient populations. Ms. Kurz has actively participated in hereditary cancer research and has numerous peer-reviewed publications, and is a frequent speaker on genetics at various venues and conferences. She holds ABGC certification and is licensed in California. She received a Bachelor's of Science in Genetics from the University of Western Ontario and has an MS in Human Genetics from Sarah Lawrence College. So as you can see, she's very well qualified, and we're thrilled to have her tonight. Please help me welcome Ms. Kurz.
Thank you. If you see me grinning from ear to ear, infrequently, it's because my family is for the first time listening to me giving a speech. So my kids and my husband are over there in the audience. So if you see me looking over there at any time, you will know why. Thank you very much for that lovely introduction. It is really my honor and a pleasure to be here tonight. I have been, as I said, as you heard, a genetic counselor for a long time, and I still believe that genetics has the power to change lives. and to save lives. And I love what I do. So every morning I get up and I enjoy going to work. I am going to talk to you guys for about, I think, over an hour, almost 90 minutes, but we will take breaks in between for questions. That's because I have a lot of ground to cover and I have a lot of things that I want to talk about. And I'm going to go over an outline with you, but there are also cards that you should, as I talk, whenever you have a question, just write it down, and then we will answer them during the question and answer period. So let's start with the outline, because so that you know exactly what we're going to be, what's on the menu tonight. So first, we're going to talk about genetics. Whole bunch of medical genetic jargon. I'm going to explain it all to you. I'm going to explain to you the inheritance and the multi-gene panels and a whole bunch, as I said, of nerdy words and concepts. You'll be able to be the genetic counselor to your family when you go home tonight. I promise you that. Then we're going to switch gears and we're going to drill down to different types of cancers. First, we're going to talk about genetics for breast cancer survivors. Then we're going to talk about genetics for ovarian cancer survivors. And then we're going to talk genetics for colon cancer survivors. Then we're going to stop for some questions where you write them down. And then updates in genetic testing methodologies, because things have changed a lot in the last 5 years. And then I'm going to share with you some of my interesting experiences, at least in the last 5 years. Most of them are my whoops moments, and I think they're important didactic moments, not only for me and for my peers, but also for you guys, because genetics is really not as perfect as we would like it to be. It's a science in its infancy. So I want to start with this slide, and this slide, for those of you who may remember, or you may not, is a slide that was on the subway, plastered all over the subway when I was in college in New York, and it's actually a perfume ad. And it's a tongue-in-cheek, at the time it was 1995, it's a tongue-in-cheek ad because they're actually using DNA to sell perfume. And they're saying, well, you know, your ability to smell and your interest in different perfume is actually genetic. Ha, ha, ha. Okay, I always put that slide up at the beginning because, in fact, that is now true. And it's no longer a funny joke. It's the truth. So our ability to smell is hereditary, and the types of smells that we enjoy are also hereditary. So that's just a little light humor to begin, because the next slide is, in fact, one of my least favorite slides, but I do put it up. I have a particular bone to pick with Time magazine. Time magazine has repeatedly put very inflammatory covers about genetics every 6 months or so for the last 10 years. So if you read the titles, you'll see Tinkering with Life and the IQ gene, and the God gene. And so you can imagine how those of us who actually do genetic work feel when we see something like that, that sensationalizes genetics and makes it seem unethical, challenging, and perhaps undesirable. However, there is one important cover, and it's the one in the middle, and I put it up there, and it doesn't come out very well because it's very old, and this is before the digital era. We couldn't just steal pictures from the internet, not that I do that. And it's the cover that was on Time magazine when the human genome was completed. The sequencing of the human genome was completed, and it was 2001. And we were so enthralled and so hopeful that this unbelievable discovery for medicine and humanity is going to change and revolutionize the way we practice medicine. Well, it's been a long time coming, but it's finally coming home to roost. And this, of course, is something very familiar to everyone in the audience. It's called the Angelina effect. And the idea here is that Ms. Jolie is a white crusader and has bravely taken on the role of being the one that showed us the way for personalized medicine. And to that I say, be that as it may, I'm not going to judge that, but I can tell you from seeing thousands of patients over the last 20 years that there are many people like you in the audience that have paved the way for her to be able to stand and have this Time magazine cover. These are patients who in the 90s gave me their sample for research, not knowing whether I'm going to give them results back. These are patients who believed me when I said, oh, you know, I think the fallopian tubes are also at risk, circa 1999, without any proof whatsoever. These are people who had surgery, hoping and eagerly participating in research so that she can then go on ahead and have a Time magazine cover like this. So it's to those women and those men that I usually dedicate most of my talks. Enough of the editorial. Let's get into the nitty-gritty. So Cancer Genetics 101, or how to sign like a genetics geek, because when I was in school, genetics was really a marginalized part of medicine, and a lot of times we were not very popular. Common disease is genetic. So whether we're talking about cancer, whether we're talking about heart disease, diabetes, even osteoporosis, there are genetic components and hereditary components to all of those diseases. It is no longer relegated to prenatal testing for abnormal carrier screening or serum screening. It is actually part of every disease that we know in medicine. Specifically for cancer, because this is what we're going to be concentrating on today, these are the risk factors. So we know there's family history, there's lifestyle, environment, aging, chance plays a big role, and also genetics. And this is, again, what we're going to drill down into today. This is a diagram that illustrates in a nutshell what our DNA and our genes are. So the DNA is a very long molecule. Literally billions and billions of molecules all in a row make up this very long string of basically what would look like a pearl necklace. And you have two copies of this very long string of chemicals, and it's very tightly wound up in packages that are called chromosomes. And those are very tightly wrapped up and put in the nucleus. And every cell has a nucleus. So our DNA is present in every cell of our bodies. And that's an important point, because if you think about it, a breast cell, for example, has hair cells in it, hair DNA in it, hair genes. It has eye color genes in it. It has liver cell genes in it. So the entire library of a human is found in every cell of our bodies. And I mentioned that, and again, I stress that because it's important to realize that when a mistake or what we call a mutation is inherited, it is present in every cell of that person's body. We'll get to that later on. But when we're born, the DNA, such as it is when we're born, is actually as perfect as it's ever going to be. As we age, aging being one of the risk factors for cancer, that DNA is copied literally millions of times. And that copying process is incorrect in all life on this planet, whether you're an amoeba or whether you are a human. That copying process of the DNA is incorrect. It's what aging is. And so the more we copy our DNA, the more mistakes we make. The more mistakes we make, the more chances we have that we mutate or put a mistake or introduce a mistake in an important gene. And that is how most cancer starts. Most cancer starts much later in life because it takes time to accumulate these mistakes or mutations. It's called sporadic cancer. So that type of mutation that is accumulated during an individual's lifetime are called somatic mutations. So those of you who've had cancer recently, in the last 5 years or so, may have had testing that's called somatic testing. So if you've had Oncotype DX or Foundation Medicine or a number of different tests, those are genetic tests, but they are actually looking at a different picture. They're not looking at hereditary causes of cancer. They're looking to predict behavior of that tumor, which arose due to mistakes that were accumulated during that individual's life. Now, germline mutations, which is the subject of my talk, are those rare instances where that particular mistake or mutation is present at birth. So it's present in every single cell of an individual's body. So a lot of times I get the question, well, how come if you have a BRCA mutation, how come I'm not at risk for brain cancer? And that's because, remember when I said that the cell contains all the genes for every type of cell type? That's because if you're a breast cell, then you use BRCA1. But if you're a brain cell, you don't use BRCA1. It's dormant and it's never used. So it's only the genes that are active that are important for that particular cell type. Okay, this is a famous, it's a hypothesis. I think it's more like a theory now. It used to be called a hypothesis, but it's called the Knudson hypothesis. Dr. Knudson in the 70s postulated the following. He said, if there is a mutation or an abnormality, it's present in the father's sperm. So you've got two copies of the gene, right? There's a purple line behind this yellow star here. So there's two copies of a gene. Let's not pick on BRCA. Let's take Lynch syndrome gene. Let's take MLH1. And so if there's a mutation in one of those two copies, and the other parent here is a female, the mom, has two copies, as most humans do, and there is a child that's conceived, then there is a 50-50 chance as to which one of those copies of the gene will be passed on to the offspring, right? Doesn't matter which one mom passes on because both of hers work perfectly fine. But there is a 50% chance here that the child, who, again, a female here, will inherit that copy that's abnormal from the dad. Now, what happens as that person then goes through life and continues to divide that cell, who becomes a daughter cell? Eventually, a second mutation is introduced. And the longer we live, the higher this is all probabilities. The longer we live, the higher the probability that this is going to happen is. So eventually, there will be two non-working copies of that particular gene. In that case, there will be no working gene product. And whatever that gene product is, whatever its role is in that cell, will not be done, okay? If that happens to be control of cell division, that cell can become a cancer. If that happens to be ability to eliminate certain toxins that accumulate in nerve tissue, that may turn out to be Alzheimer's. So depending on what that is, it's that 1-2 hit, the Knudson 2-hit theory, that really underlies most of what we understand about the molecular causations for cancer. So really, that's a lot of alphabet soup I just talked about there. Who is at risk for hereditary cancer? So it turns out, and can you guys see this? Because for me, it looks pretty, do you see this? Okay. They only account for a small proportion of all cancers. So, you know, it is really like looking into a crystal ball and trying to tell the future for those who want to know. Because it turns out that only about 10% of all cancer is hereditary. So we have a saying. Our saying is, all cancer is genetic, but only 10% of it is hereditary. And what that means is, in order for a cell to become cancerous, that cell and the lineage of that cell prior to that cell had to have acquired those sporadic mutations, right, that accumulate as the cell divides. But to be what's called a hereditary cancer, that's a situation where the mistake is there at birth. And only 10% of all cancer diagnoses fit those criteria. So how do we know? How do we, from it's all fine and good, but how do we empirically measure that? How do we identify those people who have that hereditary predisposition that's inherited? Well, people who are young at diagnosis, so breast cancer under the age of 45, colon cancer under the age of 50, rare tumors such as ovarian cancer, male breast cancer, pancreatic cancer, and so on, sarcomas in children, multiple tumors. So these are individuals who, for example, can develop breast cancer, are treated and are cured, and later on, often decades later, develop a second new primary. That's not a metastasis or a spread of the first tumor, and it's not a recurrence of the first tumor. This is a second new cancer. It's almost like lightning striking twice. Family history. That is a very important clue. What does the family history tell us? So this is where we're going to spend a bit of time because it's so important. Family history is the first genetic test. So years ago, before the genes were discovered, there were women who underwent prophylactic bilateral mastectomies simply because the mom had breast cancer at the age of 40. We didn't know if there was a predisposition there. We didn't know if the predisposition, had it been present in the mom, was passed down to the daughter. Remember, it's 50-50. We didn't know. So it really was our first genetic test taking a family history. What we've learned is that what's special about family history, and it's different than most other medical tests and procedures, is that it reflects consequences of genetics as well as shared environment and common behaviors. So the family history, which is also what we sometimes call the genetic biopsy, is really our way of understanding what risks may be present in a particular family. How do you take a family tree? So this is a class every physician should have taken. A lot of them don't remember, but you should take an accurate and full pedigree. Needs to be 3 generations, and it needs to go to third-degree relatives. So what does that mean? Okay. Here is the patient. Sister, brother, mother, father, all are first-degree relatives. That's why they have the number 1. You share 50% of your DNA with your first-degree relatives. They're your closest DNA relatives. Uncle, aunt, grandparents are second-degree relatives. You share 25% of your DNA with them. First cousins, aunts or uncles of your grandparents, and so on, are third-degree relatives. An accurate and full pedigree that can be used for risk assessment needs at least third-degree relatives. So you don't have to go back all the way to amoeba, but it would be useful to, as you're thinking about this, to ask family members, what did grandma so-and-so die of? Ask the question, because a lot of times the information is there, it's just not communicated. And so accuracy is a very important hallmark of an accurate risk assessment. Accurate pedigree is an important hallmark of an accurate assessment, because if somebody says, Grandma Jean had abdominal cancer, well, she could have had cervical cancer, she could have had endometrial cancer, she could have had stomach cancer, she could have had colon cancer, she could have had a whole liver cancer, pancreatic, she could have had so many different types of cancers, and they all have different meaning from the genetic standpoint, because different genes are associated with all those different organs. So ask the question. This is what a sporadic family history looks like. So when you take a family tree, you find someone who has colon cancer, later onset, not much of a family history. The risk for cancer of this person, or this person, the children, or any of these people who may be asking, is actually average. It's no different than the general population risk. This is a hereditary family history. Notice 50% of the people in this pedigree are what we call affected, i.e. have signs or symptoms of a disease or a condition that's genetic. Breast, ovarian cancer, prostate cancer, breast cancer, early onset 34, early onset 44, early onset 59, borderline, and this is late onset. So the important part of this here is that you notice that 50% of the people in this family tree have what would be considered the condition being hereditary breast ovarian cancer syndrome, also called HBOC. So don't forget to look at both sides of the family. That's my other pearl of wisdom from this, because if you look at this, do you notice where this is coming from? Who's it coming from? The father. That's right. This is I have 2 soapboxes, typically. This is my first soapbox. 50% of women who have an increased risk for hereditary breast cancer and ovarian cancer get it from their dads. 50% of the time. So we as clinicians are missing 50%, at least 50% of the people if we don't ask about the dad's family history. And the dad's family history oftentimes can be a grandma or an aunt. Or if dad only has a brother, it could be his father's sister and her descendants. That is why taking a thorough pedigree is so important. Because you don't want this to happen. This is a patient of mine. This happened to me. So here's a lady, and I hope this came through when I was playing with the slides today. Here's my patient, okay? No, this is my patient right here. Lady had ovarian cancer at 39. Her sister, she tested positive. She had a mutation in BRCA1. This is her sister, unaffected, doesn't have cancer yet, also positive. This stands for bilateral salpingo-oophorectomy. It means this lady, who's now 69, had her ovaries and her tubes removed at age 35, right after she had this lady right here. And then she has all these people with breast cancer, right? So you'd think, oh gosh, this is a really bad family history. So she must be the person who's carrying this. Now, she had her ovaries removed, so she couldn't get ovarian cancer. And so this is where it's coming from. Well, guess what? This is one of my oopsies back way in the 90s. We tested her and she's negative. She doesn't have it. She does not have, she's not the one that gave the mutation to her daughters. He did. And guess what? This lady right here, if she has a primary care doctor or a primary clinician who's not asking the family history all the way to her cousin, would not know. She would go on and develop, she has a, let's see, to a 25% chance of being a carrier of a BRCA1 mutation and would never know it. Told you I'd share some oopsies with you. This is what my family tree looks like, those people up there. Trouble, 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 trouble. But seriously, family history is so important that ASCO, about 3 years ago now, came out with a recommendation, which is basically their way of saying to oncologists, you must do this. They have to actually take a family tree on all their patients. Require cancer risk assessment by a qualified genetics professional, but have to have genetic tests. They have to have a family tree. So everyone's taking it seriously. Okay, so what are the other tools for estimating cancer risk? We talked about family history. Let's talk about genetic testing. Okay, so genetic testing is underutilized. Those people at council probably know that in the back. But the truth of the matter is that for many years, testing was not done in the U.S. It was done elsewhere in the world, but not in the U.S. It was because it was under patent, and I'll get to that story in a little bit because I have one of my little stories to tell you guys. But it turns out that fewer than 10% of BRCA mutation carriers have been identified. So there's people walking around, probably with a family history on the dad's side, who don't know they're BRCA carriers. Now, why is that important? Well, it's important because those BRCA carriers who may not know they are carriers have a 50 to 80% lifetime risk for developing breast cancer. 50 to 80% lifetime risk for developing breast cancer. And there's effective prevention and surveillance that is available. 35 living breast cancer patients carry BRCA mutations, but only 30% have been identified. So even those of you who may have had breast cancer, who may or may not have had genetic testing, may wish to see a genetic counselor or talk to your clinician about additional testing, because we've only begun to scratch the surface. So when should we do genetic testing? Well, we used to have some hard and fast rules, and I'm going to go over them with you, but my personal mantra is anyone who wants genetic testing should be able to have it. But these are the official guidelines. Patient has a reasonable likelihood of carrying an altered cancer susceptibility gene. So if you have no family history and you're 20 years old, their guidelines say that you shouldn't have genetic testing. The genetic test has to be available, and it has to be able to be adequately interpreted. So you could do a genetic test. We can do lots of tests, but they may not be meaningful clinically. So what does that mean? That means if you come see me and you say, oh, I have a mutation in the TIF1 gene, I'll say, oh, great. I don't know what that means. Results have to influence medical management. This is a big one, especially if we're talking about genetic testing being covered by insurance companies. They want to see that if they're going to pay for that test, which used to be $4,100, they want to see that the clinician that's ordering the test or someone else will change your care. So that test is then useful to you somehow. It's not just informational. It's not a party trick. Like a 23andMe, which is a company that does direct-to-consumer genetic testing. We can talk about that later if we have time. That's not what they want to see. They want to see that a test that is going to change the way somebody is going to take care of you. And then, if you want the information, because you have to remember, not everybody wants this, right? Some people don't want to know, and that's okay. That's okay. But if you want to know what changes you can make before a diagnosis or how you can change your treatment and tailor your treatment, then genetic testing is part of the answer. Why would you want to know? Well, personal risk to get cancer or another cancer. My children's risk to get cancer. Oftentimes, and we'll talk about this later on, it's important to test a person that has the signs or symptoms of whatever condition you're looking at, whether it's cystic fibrosis or Tay-Sachs or Huntington disease or whatever it may be in genetics. We want to find what that person who has the signs or symptoms of the condition has, if there's a mistake there, and then see if anybody who is related to that person has inherited it or not. Maybe your sister wants to know what her risk of getting breast cancer is. And the important part of it here is, again, and I keep drilling this in every slide, it feels like, but screening and prevention are often available and may save a life. So it's a multi-step process. It used to be, at least. It doesn't need to be like this anymore. Again, a lot of these slides are sort of the status quo that our society guidelines tell us that we should follow. I'm a little bit of a rogue person, though, and I'll talk to that a little later on. But you have to identify who's at risk. And then you have to provide pre-test counseling. So why are you doing this? Then you have to obtain informed consent. So that's part of it where you say, this test is going to mean this. Do you want to do it, yes or no? And then you order the genetic test, and then you disclose the results, and then you provide post-test counseling and follow-up care. And all of these societies, and this is alphabet soup of societies, actually recommend those exact guidelines that I just told you. Now, I'm going to step aside for a little bit and tell you what I really think, and that is that genetic testing, in order for it to be accessible, has to skimp on some of those steps. So perhaps, the pre-test counseling is not a one-on-one session. Perhaps for that particular individual, they attend a session like this. And that's considered a pre-test counseling session. We just talk about general genetics and you understand what it means. And then you go and see your doctor and they do a quick family tree and then you draw the blood. So there's many different ways of trying to identify who's at risk. And we as a genetics community, and I think I'm the only one in the room, so I can say whatever I want. Oh, actually, no, there's a genetic counselor in the back. She'll tell on me. But we in the genetics community have to be flexible and have to understand that the way we get information out to you guys may not be in the way that we think it should be, as long as it's getting done, okay? All right, that was soapbox number 2. So there are definitely very clear recommendations that exist. These are general. These are not specific to any one gene when somebody tests positive. So if you have a positive test result, test the other relatives, all of them. All of them. Your cousin, third cousin once removed, if you can find him or her and tell him or her about the mutation in the family, please do so. You probably will save his or her life. Now, for the person who's had the test, so the proband, there's increased surveillance if the person hasn't had sign or symptoms of the disease. Lifestyle changes. There's chemoprevention. That's our name for the idea that someone can take medication to reduce risk. And then prophylactic surgery. So the best person to test, as I said before, or I go back to this slide, is really the person that is different, right? That has the signs or symptoms. So if we're looking for a gene for yellow apples, you would test the yellow apple, right? Not the red apple, right? That's the thinking behind why it's so important to test the people who've had the cancer in cancer risk assessment. So pearl of wisdom number 3, when searching for something rare, which is a hereditary cancer, start the search with the most likely target. Now I'm going to switch gears a little bit, and I'm going to talk very briefly about the types of genetic test results. I don't know how many of you guys in the audience have actually had genetic testing. So what I'm going to do is very briefly go over the 3 types of genetic test results that you can get. But before I do that, you need to understand how this works, because, and this is a very famous genetic little ditty. It's called The Big Red Dog Ran Out. And if this is a gene, this makes sense, right? It has a beginning, an end. The sentence makes sense. It's a full sentence. Oftentimes, the mistakes that occur as we age, which are part of the normal aging process that I talked about before, are the ones that are called missense. So all of a sudden, one of the letters is changed from an E to an A. It's called a missense mutation. So let's read that together. The big red dog ran out. Well, we're in California, and that makes sense to us. But if we were in, I don't know, Zimbabwe, that would be something that doesn't make sense. And this is a perfect example of what human variation actually is like. A lot of times it's dependent on context. So there's a famous story in Australia of a geneticist who was talking to a patient. And the patient was saying, well, you know, there's such high risk for melanoma in Australia. Why is the risk for melanoma so high? And he was an good old boy, and he said, well, listen, here's the deal, okay? You know, 2 generations ago, you guys were living in the UK, and you basically saw 3 days of sun. So you got that way, you were very pale. You got that way because up there, you would break your bones if you weren't very pale to absorb all the sun's rays, or those 3 days that actually showed up. So millions, no, not maybe millions, but thousands of years of genetic evolution created individuals that are suited to a very northern type of climate, okay? Well, all of a sudden, you took those people and you put them on a boat, and within 6 months' time, you place them in a tropical climate where physically, because the Earth is round, right, you are miles closer to the sun. And you're not genetically designed to be here. So this is very similar to that. Context is everything, right? That's why the risk of melanoma is so high in Australia. It isn't because there's some ozone hole in Australia. There isn't because there's, magically increased risk in the Australian population. It's because most Australians actually are not designed for where they're living in, excuse me. Okay, so another change that can happen is when all of a sudden there's a message, the message is shortened, or the recipe or the sentence is shortened. So this is a stop code. And yes, very nice. Yes, thank you. I love audience participation. It means I'm doing it right. The most common causes of human disease, though, are these called frame shift or deletions or insertions. So what this does is it basically shifts the reading frame, because you see, although I didn't mention it, what I was doing is I was reading groups of 3, right? So the DNA is actually always read in groups of 3. So even though it's a long string of chemicals that's 3 billion base pairs long, every single time it's read, it's in groups of 3. So if there is an IG up here that's missing, all of a sudden what you get is this gobbledygook, okay? This copy of the gene is not doing its job. It's not going to make a protein or a gene product that works. That's a mistake or a mutation. Same with an insertion. If you put a Z in, everything that comes after that isn't going to work because it doesn't make sense. That's another mutation. Last thing I'm going to mention here. If any of you have heard of SNPs, they stand for single nucleotide polymorphisms. That's a SNP. Most human variation is a SNP. Just a benign change. Okay, so let's do interpret and test results. There's 3 types of results you can get. You can get a positive for a deleterious mutation. You can get no mutation detected, or a genetic variant of uncertain significance. So no mutation detected, I'm going to start with that one because that's usually the most common result, but it also happens to be the most challenging because it could mean 3 different things. Well, it could mean there's a mutation in another family member, right? Because we didn't test the yellow apple, maybe we tested the red apple looking for the yellow gene. So maybe the yellow apple has the gene, the abnormality, but we should always test the person most likely to be positive, going back to that analogy. Or maybe it's a true negative. Maybe there's a really strong family history, but remember, only 50% of the people in a family history are affected. And if we happen to test someone who is unaffected and is negative, maybe it's really true and unrelated. Maybe that person's truly negative. But we don't know for sure because we haven't found the cause of the yellow apple, right? So we can't label that person a true negative. Or maybe there's nothing going on in the family. Maybe there's other risk factors. Remember we talked about family tree and pedigrees? I showed you all the other risk factors like diet and lifestyle, all those other things that families can share. Maybe that's really what's causing the cancers in the family. So when you get a negative result, it is almost, unless are able to test somebody in the family who has the condition and is a positive for a mutation, you almost never know which one of these 3 scenarios you're dealing with. And it makes it very challenging from the clinical standpoint, because oftentimes I have a patient who's in front of me and says, okay, my result's negative, what does this mean? And I'll say, well, you know what? Let's look at your family tree, because this is really not informative. It's not helping me take care of you at all. Variant of uncertain significance. Oh, they got a lot of bad press lately. I don't know how many of you guys have had genetic testing lately, but they have gotten a lot of bad press. So what they basically are is that benign variation I showed you before. Those SNPs, VUSs, variants of uncertain significance, are changes or alterations from the expected sequence of a gene. So if we're expecting the word red and we have the word rad, that's a variant of uncertain significance, depending on context. So a lot of labs are unable to call it because it really doesn't exist. It's an abstract. It doesn't exist. It's either nothing or it's something bad, right? This in-between thing doesn't really exist, the construct. And so clinical significance is not known. We can't use it to interpret results. We have to manage the patient using their personal and family history. We go back to the pedigree. And sometimes we can clarify it by having genetic testing in other family members who are affected. There was a time when we could actually track these variants, but labs no longer do that now. It's getting pretty prohibitive from the cost standpoint. But the bottom line, and this is a take-home message, this should be my pearl of wisdom number 5, but I didn't get a chance to do it. Ignore the result. When you get a variant of uncertain significance, I want you to ignore it. Okay, you take the result, and so I do in my clinic. I turn it upside down. I'm saying I'm going to ignore that. I'm going to pretend I didn't order that. I can't use it. Not only that, but I'm not going to test other family members for it in the hopes that it's going to give them information that's going to help them manage their risk. It's incorrect and erroneous to do that. And there have been cases, some of which have been published, where patients have done that, or clinicians have done that, and patients have had surgery unnecessarily. So do not use a variant of uncertain significance to guide your care, or anybody in the families. Okay, and this is actually the simplest result. It's not the one you want, but it's the easiest one to interpret because we know what we're dealing with. Well, it's some management per NCC in our published literature. And we have lots of recommendations, and I'll go over some of them, but they're all easily accessible on the internet. The important part of this one, though, is remember there's a 50% chance of passing that on to offspring, siblings, and one of the parents is carrying it, okay? Okay, finally, the empiric data, and I make that go in and out because this really is what most of the genetic literature really is. Odds ratios and relative risk and probabilities and experiments and all of that all is actually summed up in this one slide because these are the risk factors for breast cancer, for example. Just giving you an example. Aging, early menarche. That means what age you got your period. Late menopause, if you went through menopause late. Breast density. Nulliparity, that just means whether if you've never been pregnant. Estrogen use. Dietary factors. Alcohol is actually the only dietary factor that is a true risk for breast cancer. People always ask me that. Alcohol is the one. And age at first live birth. How old were you when you had your first baby? Whether you breastfed or not, and if so, for how long. But if you look, in this particular slide, if you look at those risk factors, they're all markers for estrogen exposure. How long was the breast tissue exposed to estrogen? And not only that, but how, so the number of years, but how early in life did the breast tissue fully differentiate into tissue that produced milk? But there are other clues to hereditary cancer syndromes. We talked about them before. I'm going to repeat them here really quickly because I want to get to the rest of it. Cancer in 2 close degree relatives. This is a family tree. Earlier age at diagnosis than expected, multiple premature, bilateral or multiple rare cancers. I think I talked about that already. And then tumor types consistent with a cancer syndrome. So it turns out that certain genes predispose to certain cancers in particular organs. So breast and ovarian cancer go together. Endometrial, colon, and ovarian cancer go together. And then all evidence of autosomal dominant transmission, which is that pedigree I showed you where 50% of the people were affected, but they were affected people in every generation. It doesn't skip, cancer does not, these predispositions, they don't skip generations. That's the other thing, I hear that all the time. Oh, it skipped a generation. No, it didn't. It just didn't manifest itself. So what's my risk for cancer? I don't know. Well, it's qualitative, we could say, quantitative, we can say 1, 2, 3, 4, 35% risk, 85% risk. What people really do better with is qualitative. You have a high risk, you do not have a high risk. And so really it's a lot of times something like that. When we communicate risk, it's very challenging because it's not, humans are not, we don't process numbers in a way that helps us necessarily make decisions about our personal health. So there'll be lots of times when I will be talking to a patient, and she'll just say, okay, just tell me yes or no. That's all I want to know. Just tell me yes or no. And that's part of the challenge because I can't say yes or no. Well, I can say yes or no with Huntington disease, but that's not part of this discussion. Huntington disease is 100% penetrant, so for that I can say yes or no, but it's extremely rare. Okay, let's get through some of this. There's, Miriam's much more versed in this and able to probably hold an entire course in this, but there's obviously very clear concepts that will help an individual make an informed decision about risk perception and management choices. Very quickly, this is a non sequitur, but I don't have another place to put it. People ask me about insurance discrimination. So GINA, which stands for Genetic Insurance Non-Discrimination Act, is something we fought for very, very hard in the genetics community. We lobbied senators and everyone that we could would listen to us for over a decade. And finally it was passed in 2007, I want to say. And the purpose of the legislation is to protect the privacy of Americans. It's federal legislation, so it applies in every state. And it's really to prevent genetic discrimination in employment and health insurance on the basis of genetic screening or genetic testing. So, 2009, look at that, I was wrong. But it defines, and this is the important piece, it's a piece we fought for a long, long time. It defines genetic information as predictive genetic tests. So if somebody has a BRCA test and hasn't had cancer, that's a predictive test. Family members' genetic tests, so it protects the family and family history information. It also protects the pedigree, which is huge, because remember we just talked about all these results that are meaningless, and then we use the family tree as the one tool to guide care. Well, that turns out to be protected as well. And they apply to group and individual health insurance as well as employment practices. However, it does not cover, GINA does not cover life, disability, long-term care, and other forms of insurance. However, in California, there is state law that covers disability, long-term care, and employability. So in California, we're doubly covered. So who will get what? Genetics is the key, and yes, sometimes I do feel like a genie that's peering into the box. And this is where I think I probably should stop, even though I originally had planned to stop somewhere else, but I probably should stop here and ask you guys to write the questions that you have so far and hold them up when you're done. You don't have to be done now. You can be done later. But once you're done, just hold them up so that they can be collected and they can be answered at the end of the session. Okay, let me get a drink of water. I'll be right back. Okay. So, genetics for breast cancer survivors. And this is where I want to tell you guys a bit of a story. This is a family of women. These are some of the first women where it was recognized that breast cancer was inherited. And this is the mom and the two daughters. And they were actually in one of the Time magazine articles on BRCA. But I put them up here because they really were the pioneers in this. And their fight was really picked up by a woman called Mary Claire King. And if you haven't heard of her, and you have some time on your hands, I highly, highly recommend seeing a movie about her life and about the discovery of the BRCA gene. And it's called Decoding Annie Parker. And the discovery of the BRCA gene really was and reads as a Hollywood story. There were two groups of individuals, both internationally based, one in Salt Lake City, another one in France. And they were racing in the 90s to try to find the BRCA gene. Their leader was Mary Claire King. She happened to be with the Salt Lake group. Now, Mary Claire King is a remarkable human being, and she was the first person to get an NIH grant to demonstrate that breast cancer is actually can be an inherited condition. And she went back to the NIH for it sounds like over 10 years, saying, I think, breast cancer is hereditary. And they would say, no, I don't think so. No, you're not going to get you don't get any money to study this. So she collected a lot of the families on her own time. And the story goes that so it came about that they got very close, and both groups were very close to discovering the BRCA gene. And the one group that actually won was the Salt Lake City group, whereupon they promptly turned around and patented the gene. Now, how they patented something that exists in every single person in this room, every single person in this room has two BRCA1 genes, all of us. How they were able to patent that, I will never understand, but they did. And then BRCA2 was shortly discovered after that, and they patented that too. And I want to say 2 years into it, they put every lab that was doing genetic testing out of business, enforced the patent, and raised the price. And from 1998 onwards, the cost of genetic testing was outrageously high. When I got out of school, it was 1997, and a BRCA test cost $1,800. By the time the patent was struck down in 2013, that price had skyrocketed to $4,100, which is why so few people have had genetic testing. So when it was struck down, a whole bunch of companies, council included, raced to create much more affordable testing. And that is where we are today. But the story, I'm telling you this story for two reasons. The first is because genes are not patentable. That's like saying if I take out my tooth and I give it to you, is that an invention? And you hold it in your hand, is that an invention? No. But it artificially depressed the ability for us as clinicians to identify who is at increased risk. And it has cost women cancer diagnoses, and some of them their lives. And that's, so box number 3. I know I said there's only 2, but there's really 3. These are the 3 women from, these are these women that I talked about. This is them when the gene was discovered. It was 1993. I won't tell you anymore. If you get to see the movie, do see it. It's really remarkable. So this is what a hereditary breast and ovarian cancer looks like. I've shown this before. You could see there's 50% of the people have the condition, in this case, breast and ovarian cancer. But that's not all of it, because it turns out, and I don't know if you can read this from over there, that while this is a hereditary type of 1, 2, 3, 4 pedigrees, there are other types of pedigrees, what we call familial clustering. So it's not as much cancer as we see here, but a lot more than we would see in the sporadic case. So these are the 3 types of family trees we see with BRCA and with breast cancer. So in reality, if you look at breast can you guys still hear me okay? If you look at breast cancer diagnoses, how much of those are inherited? So about a quarter are what we call traditional inherited breast cancer. Most of them are hereditary, and that's that sporadic cluster, which we're now beginning to unravel, by the way, with multi-gene testing. We used to say this was a black box, and I don't know, but I think more and more we're able to quantify what this means and what genes are involved here. Some of you may have seen these slides if you've ever gone to these talks before. These are the risks for BRCA1 and BRCA2. So a woman who's had breast cancer before her diagnosis had a risk of 50 to 84% for developing breast cancer. The lower risk is for BRCA2. The higher risk is for BRCA1. Then there's a risk of ovarian cancer, prostate cancer. Is that still working? Okay. I don't see it from my angle. I apologize. Pancreas cancer and male breast cancer, there is a risk of that between 7 to 9%, depending on the study. The risk for pancreatic cancer is lower in women, but even so, it's considerably increased compared to the general population risk. Prostate cancer risk is the highest risk in males who are carriers of BRCA1 and 2, mostly, usually, and that's a 20% lifetime risk. So it's just another way of showing the same information. So prior to a diagnosis of breast cancer, the likelihood for developing breast cancer was about 87%. That's compared to the general population risk by age 70, which is about 12%. And the risk of breast cancer by age 50 is 50%. Ovarian cancer risk is 54%. Again, they take the BRCA1 risks because those are higher than BRCA2. But once you've had breast cancer, then what, right? Finally here. Took us an hour, but we're finally here. So once you've had breast cancer, then what? Well, it turns out there's a second breast cancer risk, and that risk at 5 years can be up to 27% in BRCA carriers. So this is a situation where you develop breast cancer, you treat it, and you're cured. And then 5 years later, there's up to 27% chance during those years of developing a new second breast cancer. Not a metastasis, so not a spread of the first, not recurrence, but actually a second new event. By age 70, in BRCA1 carriers, that risk is 64%. I cannot tell you how many women I've seen in my clinic who will say to me, "You mean to tell me that I had a risk of getting a second breast cancer that was 64% and nobody told me? Nobody gave me the option of doing genetic testing, because I would have done a bilateral prophylactic mastectomy on the other side if I had known that there was a risk of 64% of this happening again by the time I'm 70. I don't want to go through chemotherapy again. I don't want to go through surgery again." So it is crucial and important that genetic testing is available to women at time of diagnosis, and that's so box number 5. I have a lot of them. Because that's when you need it. You need it when you're diagnosed. It's useful later on as well, but at time of diagnosis is when you need it, because that's when you're making decisions about how to treat the cancer and also, ideally, how to prevent this from happening again. Should you be one of those rare people that has a BRCA mutation. Okay. This is just an example. I'm putting this slide up just because I wanted to show you guys the difference. The breast cancer rate of rise, you could see it's pretty much you know, from about age, here we go, about age 30 on, it just continues to climb up and up and up and up, okay? Ignore the ovarian part. I couldn't take it off the slide. We'll talk about ovarian in a little bit. The other question I get all the time is, well, what's my risk now? I'm 46. What's my risk right now? So this is a meta-analysis that was done by Parmigiani and Chen. It's a little dated, but it's still very accurate, and it shows you, it looks a little unclear, I don't know why that is, but it shows you the risk of developing breast cancer by a particular age. So by 50, right, it's 29% and 21% if you're now 20. But if we're talking and you're 40, and you want to, if you're 40 and you say, what's my risk in the next 10 years, that's 15 and 20%, depending on which gene it is. So the data is out there. If you need that information, that data is out there, and I can have this table available to you guys afterwards. These are the management guidelines for BRCA carriers. I'm not going to go into them in great detail because they're well published. They're all over the internet. There's a lot of small writing, but just some highlights just to kind of talk about this. The important part of it is, I don't know where this is. I'm sorry, I can't see where this is pointing. But what the important part of it is breast awareness starting at age 18. So what does that mean? That means clinical. Means breast self-exam, breast screening, age 25 to 29, annual MRI, mammograms, not till age 30, ideally. So MRIs are better in young breasts. And then discussion of risk-reducing mastectomy. Chemoprevention is another option. Chemoprevention for breast cancer is the idea that you would take a medication to reduce your risk. That medication typically is tamoxifen. So tamoxifen has been traditionally used to treat breast cancer since the 70s. It's very effective in ER-positive breast cancers. The idea is that you would give somebody tamoxifen to reduce the risk of developing breast cancer. It's chemoprevention, that's another option. So for every, there really are three options. We saw those branches over there, right? There's chemoprevention, preventive surgery, and increased surveillance. And again, all of these are I can make available to you guys if you want to read them in depth. There's a risk for men too. So, and this is an important part, especially because if we talk about brothers, sons, fathers, uncles, cousins, etc., the risk for prostate cancer is 20%. It's more than almost triple that of the general population risk for getting prostate cancer. There's a risk of pancreatic cancer that's significant, and then there's a risk of male breast cancer, especially in BRCA2. There is such a thing as male breast cancer. I see some surprised faces. But there is such a thing, and it's more common in BRCA2 than BRCA1. I mean, the probability is still less than 10%, but in the general population, that risk is something like 1 in 1,000 or something. So it's increased many times compared to the general population. Okay, so there's management guidelines for men too who are BRCA carriers. Keeps on going. Again, I won't go through all of this because there's a lot in there, but I'm happy to share these with you guys, and you can digest them at your own leisure. So again, if we haven't done this already, it's time to write some questions down. You can use more than one card. I think there's some cards on some of the seats that nobody's sitting at. And you just hold them up, and I won't be offended if you're holding up the card while I'm speaking. So now ovarian cancer. So genetics for ovarian cancer, because we're going to hit breast, ovary, and colon. How much ovarian cancer is hereditary? Well, I think you guys by now can probably tell me, because this is this repeating pattern here, right? About 12 to 30%. Okay, that slice is not quite 30%, it's a little less. But about 12 to 30% of it is hereditary. And it turns out, and I promise this is my last soapbox, number 8 I think we're at, 12 to 30% of all ovarian cancers are inherited. It is the highest probability of a BRCA mutation or any genetic predisposition to cancer of any cancer, any cancer. And as such, it is imperative that anybody who's had ovarian cancer has genetic testing. And most societies have guidelines to this effect. but I'm not sure that it's actually trickling down to routine clinical practice. So if you have had ovarian cancer, if you know somebody who's had ovarian cancer, make sure they get genetic testing. Number one, the likelihood that they have a mutation is the highest than any other cancer. Number two, there are options for chemotherapy that are available to BRCA carriers, especially women with ovarian cancer, that they may be able to avail themselves of. And number three, and importantly, finding those women who have ovarian cancer because of something that runs in the family can save the lives of the other women in the family. And that's really important. It's you guys who really make the difference, because you'll talk to your friends and you talk to your neighbors, and a lot of times that patient ends up in their oncologist's office or primary care doctor's office because of something that you guys said. So again, soapbox number 8, everybody with ovarian cancer needs genetic testing. Well, what are the risks? They're just a reiteration of the other one, but I do have something extra, so just bear with me. So the breast cancer risks in BRCA carriers, these are women who have had ovarian cancer, okay, is 50% by age 70, 87%, we already know that. But it turns out that if a patient has had ovarian cancer, at 5 years, the risk for breast cancer is 27%. So in those 5 years, she needs breast MRIs, she needs discussion of chemoprevention. There are other things that ovarian cancer survivor needs other than other than follow up and care for the ovarian cancer. So it's important to know whether there's a predisposition there. The breast cancer by age 70, look at that, it's 64%. Again, pretty high. This is that same slide, only this time ignore this part. What I want to show you here is the difference between BRCA1 and BRCA2. BRCA1 and BRCA2 act very differently for ovarian cancer. They act very similar for breast. This is for both breast and BRCA1 and BRCA2 here. But when it comes to ovarian cancer, look at the difference. This penetrance, or the likelihood of developing cancer for BRCA1, is about 54%. BRCA2 at its highest is about 27%. Look at the rate of rise. And again, this is a very important piece of the puzzle when we talk about how do we take care of people who have had breast cancer and we need to worry about their ovaries. Look at that rate of rise. It starts around age 35, and it really steeply climbs from there to about age 60, and after that it kind of tapers off. Whereas BRCA2 kind of just moseys along, and around 50, that's when it really starts to rise. So there's a difference on how it presents. Again, it's important to remember when you're making decisions. This is a meta-analysis of a very same article, just looking at a different cancer site. It talks about the risk of cancer based on the gene and the decade of life. Again, I can have these available to you guys afterwards if you want to go through them more meticulously. And it's going to become a common refrain. This is the NCCN guidelines for ovarian cancer. It includes ovarian cancer management and breast cancer management. And this is where I think it's not as routine, because a lot of times people don't think of ovarian cancer and Lynch syndrome. And this is my segue into my last piece, which is the colon piece. But Lynch syndrome is or also known as HNPCC, hereditary non-polyposis colon cancer. That's a mouthful. Say that 3 times fast. That's why we call it Lynch. It's called Lynch because Henry Lynch is the geneticist that first characterized it in the 1960s. Anyway, these individuals present typically with colon cancer, but there is an ovarian cancer risk. So when you're looking at a patient who has had ovarian cancer, BRCA testing is not enough. That's not enough. And so many times I see it where people will come and say, well, I had BRCA testing and I'm negative. And then we do a panel test, which is what we're going to talk about later, where we kind of do a buckshot approach for trying to find out genetic mutations. And lo and behold, we find a Lynch mutation. So ovarian cancer is part of Lynch syndrome. Genetic testing for Lynch syndrome should be part of the genetic testing regimen for somebody who's had ovarian cancer. The other reason it's important is because oftentimes with Lynch, you get what we call synchronous endometrial cancer. and that is endometrial cancer and ovarian cancer at the same time. And oftentimes that's not detected till the pathologist slices the specimens very thinly after surgery. And a lot of times people will go in and say, well, I had some bleeding, and they come, there's endometrial cancer, and that's the first diagnosis. And then later on, when they do additional studies, the diagnosis of ovarian cancer comes back and says, oh, and by the way, guess what? There's ovarian cancer too. So Lynch syndrome is very similar to ovarian cancer syndrome in that for ovarian cancer, we talk about removing the ovaries and the tubes. And that's something that women with Lynch syndrome should consider. I don't know if this came through very well. I can't really read it on my end. So let me turn around here and see. This is a slide just to show you, and I'm just going to quickly point to where you need to look. It basically shows that there's a particular type of ovarian cancer, what's called endometrioid right here. This particular cancer is typically very rare. So when you look at the types of ovarian cancer that people get, this is something like 1% of the population, 1% of the ovarian cancer, excuse me. In Lynch syndrome, it's almost 13, almost 20%, so it's increased. So whenever we see an endometrioid ovarian cancer, it's very important that we think about Lynch. All right. NCCN guidelines for Lynch syndrome, managing the ovarian cancer risk. You can see there's very dense, but basically what it says is for ovarian cancer prevention, remove the ovaries and the tubes. For colon cancer prevention, annual colonoscopies. For upper GI cancers, which are part of Lynch, depending on the family history, upper GI series, and for endometrial cancer, remove the uterus. So a lot of times for Lynch carriers, which is different than BRCA, the recommended surgery is a complete abdominal hysterectomy, so removing the uterus, the tubes, and the ovaries. Okay. A new entity. This is really kind of popped up in the last, I want to say, 5 years or so. And it's really through these multi-gene panels that I've been able to characterize it, is this idea of site-specific ovarian cancer. I made that up. That's not a real term. You won't find it anywhere else. But what it means is that you have a family history of only ovarian cancer, and the genes that cause it are here. And the importance of these genes is that oftentimes when you have somebody who's ovarian cancer and she's BRCA negative and she's Lynch syndrome negative, she may actually have one of these other genes. So which is why, again, multi-gene panel testing that we're going to get through right away, pretty soon, is important. And this is just, again, what's becoming now the routine NCCN guidelines for looking at ovarian cancer. OC stands for ovarian cancer risk in some of these genes. Okay. Do you guys have any questions so far? I'm about to move on to colon cancer. Okay, you have a whole bunch? No, I'm about maybe 10 more minutes. Okay. Is that good?
Yep, and at the end we'll collect the last round of questions,
okay.
And then I'll ask you these ones.
Perfect. Thank you. Okay. Coming down to, that's the finish line. All right. So colon cancer is divided into whether there's lots of polyps or whether there's none or a few. And we call that polyposis when there's lots of polyps. And then when there's no polyps, we call that non-polyposis. And when there's some, we call that oligopolyposis. You don't need to know the rest of it. You just need to know that colon cancer is typically divided in the ones with lots of polyps and the ones with very few. A very similar slide. Again, about 30% of it is hereditary. Let's talk about the non-polyposis one first, because it's the Lynch one, okay? HNPCC or Lynch. The early onset is less than 45. So CRC stands for colorectal cancer. Colorectal cancer less than 45. Proximal colon predominates, but distal lesions not excluded. Proximal colon is your right-sided colon. I don't know. It's this part right here. So the ascending colon. This is your transverse, this is your descending. Extracolonic cancers, endometrium and ovary, What do you know? Stomach, urinary tract, small bowel, a whole bunch of other ones with very low penetrance, about less than 10% for all of them. And these are the risks. And this is a blue slide because I could not figure out how to change these axes to black, so I had to make it blue so you could see what I'm saying. But this is the lifetime penetrance for the different types of malignancies in HMPCC. So colorectal cancer is the highest. It's about 80% for both men and women. Endometrial cancer is anywhere between 43 to 60%. Stomach cancer, biliary tract, and ovaries 9 to 11. So that's the likelihood. If somebody has HMPCC, also known as Lynch, this is what they're dealing with. Family histories, oh, that didn't come out. Well, it's a family tree. 50% of the people are affected, only this time with colon and ovary and endometrium. But this one probably comes out a little better. This is a hereditary family. 50% of people are affected. Multiple primary cancers, multiple generations affected. Not a family, not a hereditary family or a sporadic family. Remember going back to the beginning about an hour ago, we started talking about sporadic versus hereditary. On your left you have sporadic, on your right you have hereditary. The genes involved in Lynch are called mismatch repair. So if you hear that term, you don't need to know. Basically their job is to correct errors. Remember how I said we make errors as we age and as part of normal aging? Their job is to correct some of those errors. They can't correct them all because Mother Nature is not perfect, but their job is to correct as many as possible. So when the editor or the corrector of the errors isn't working, then more errors get by with subsequent generations, and that whole process is accelerated. A microsatellite instability, you don't need to know this, I'm just putting it up there because you might hear the term, is a term we used to use for this very particular type of test. And it's a cardinal feature of Lynch, or HMPCC. It's present in 90% of tumors that are caused by this particular syndrome. And if somebody has colon cancer, they do the MSI test, and it's high or positive, that means it's almost diagnostic for Lynch syndrome. Now, the sad news is that we don't use that anymore. They don't use MSI anymore. It's very expensive and very complicated to do. So now they do IHC. Now, IHC, and Ann and I had this discussion earlier this week, is not MSI. People say it is, but it's not. And it's apples and oranges. They're both fruit, but they don't replace each other. IHC is the idea of creating an antibody that attaches to the end of the protein of whatever you're looking for. In this case, those MMR genes. And if the correct end is there, right, so nothing's truncated or shifted or deleted or missing, if the end is there, then it attaches, and IHC is positive. So that means that tumor has working gene product of whatever gene we're looking for in it, okay? Most of the time that means that it was caused by not a hereditary alteration, but sometimes the alteration is in the middle of the gene, right? It screws up the middle of the gene, screws up the middle of the gene product, and all of a sudden you have an IHC that's normal, but a mistake that's present that's interfering with how the gene product is working. So it's not foolproof, and what you need to know is that IHC is not the same as MSI, and when somebody says positive for MSI by IHC, you can say that's not true. They're not the same thing. Okay, diagnostic guidelines for HMPCC, there's a whole bunch. There's Amsterdam criteria and Bethesda criteria, and they're really named based on the city that the researchers really wanted to go party in and talk about and create these guidelines. So Amsterdam were the first ones, and then Bethesda were the second ones. You don't need to know them, but they're there in case you're wondering why somebody's using those terms. There's a whole bunch of genes involved in Lynch. BRCA is much easier because there's only 1 and 2, but Lynch has about 5 genes really that are involved in it. MLH1 and MSH2 is the most common. They're most common two. They cause about 80% of Lynch syndrome cases. MSH2 has these large mutations that are very hard to pick up by routine genetic testing. So make sure the lab, if you're doing Lynch testing, make sure the lab actually has the ability to pick up these mutations. Otherwise, you're getting a false negative result. MSH6 was discovered in about 2000, I think. And what's interesting about MSH6 is that it usually presents with endometrial and ovarian cancer, not colon. So the highest probability of cancer with MSH6 is endometrial, followed by ovarian, followed by colon. So it kind of flips all those probabilities. And MSH2 and MSH6 bind together. You don't need to know that, but it's interesting. And MLH1 and PMS2 bind together. That's what I mean by a heterodimerized. They bind together. There's a founder mutation in MSH6 too. You don't need to know anything about it other than the fact that it's make sure that lab tests for it. It's very hard. It's what we call in genetics a hard to do. And so make sure that the lab tests for it. Oh, look, we're talking about MSH6 here again. Apologize for that. MYH. MYH is, I see this a lot on people's genetic test results, so I want to spend some time on it. MYH is a recessive gene. It is unlike any of the genes we've talked about so far. You need 2 non-working copies to show signs or symptoms. So far, remember, everything was 1 working, 1 non-working, 1 working, 1 non-working, right? 50% risk from the parent. MYH, in order to show signs or symptoms, you need to have 2 non-working MYH genes, 1 from each parent. So when somebody gives me a genetic test result and says, oh, I have an MYH, I'm heterozygous, which means I have 1 working, 1 non-working for MYH, I will say, okay, that really doesn't increase your risk for colon cancer significantly. So that's why that's there. It is, well, that's what, when I say biallelic, that means 2 mutations, or homozygous, that means 2 mutations. Ah, look, I animated that. Recessive inheritance is the take-home. Endometrial cancer we talked about already. In some families, the endometrial cancer can reach 70% lifetime risk. So it is important that if there's any, in women with a family history of colon cancer, if there's any unexplained bleeding post-menopause, that's worked up immediately and taken very seriously. We won't go over that, but NCCN has guidelines for colorectal too. And the importance of this is colonoscopy is the only tool that we have in cancer genetics that's truly preventive for cancer. So when you have a colonoscopy and there is a polyp, and that polyp is snipped and taken away, that removes the cancer risk. And so it is for people who adhere to the colon screening protocol, which is not easy to adhere to, being every year, the likelihood of developing colon cancer is almost eliminated. So you have a condition where the penetrance of the cancer is 80%, and then if you do the screening, you almost eliminate the likelihood of developing that disease. All right, polyposis syndromes, very fast. These are the ones with lots and lots and lots of, and that's my gross slide of the day. Lots and lots of polyps. This is not 1, 2, 3, 4, 5, 60. This is 600 polyps. These people get colon cancer 100% of the time if the colon is not removed. It's extremely rare, but it has the highest rate of de novo mutations of any genetic condition. It's a huge gene, it's commonly mutated, and we often see it in colon cancers when there's no family history at all. So when there's no family history whatsoever and you have somebody with 150 polyps at the age of 28, they probably have FAP. And it didn't come from their parents. It just happened to them during their development, their embryogenesis. And so that's what de novo means. That means it's not in the family above them, but it can be in their siblings, and it can be passed on to their children. So characterizing individuals with early onset colon cancer in the absence of a family history is very important because of this high de novo rate for FAP, and that's a take-home message for FAP. Okay, there's other risks with FAP. They're super, super rare and low. The hepatoblastoma is by age 5, which is a childhood cancer. And then there's these other chirpy, was a congenital hyperplasia of the retinal pigmentosa epithelium, blah, blah, genetic alphabet soup, but it's increased and super rare otherwise, but common in FAP. Attenuated FAPs, later onset colon cancer, fewer adenomas, same gene. We don't know why sometimes it presents with fewer adenomas, but it does. So we have a name for it. There's guidelines for it too. And for the attenuated FAP, So with colon cancer, the bottom line is, do you have polyps? 10 plus, these are the genes that cause it. You don't have any polyps? Under age 50, right-sided lesion, these are the genes that cause it. Okay, so I lied. I said I'd be ready now. But if you guys give me 5 more minutes, I'll go to the multi-gene panels, which I think is really important. It's a new technology. So the idea is that you are testing more than one gene at the same time. And the cost for doing that type of test is similar to doing one gene. So the idea is, well, if I can do 100 for the same price, why don't I just do 100 as opposed to 1? Okay? The idea there is, well, more information is good. Whether you believe that or not is really up to you. But there has been a paradigm shift in my world that has changed the way I practice. So before, the idea was, okay, you gather the family history. You gather the family history and you do a physical exam and you generate a differential diagnosis. This is our idea. Well, it could be this, or it could be this, or it could be this, but I only have $1,000 to use based on their insurance, so I'm going to go for number 1 only, and hope that it's not number 2 and number 3. So that really was how we practiced genetics for a long, long time, because we can only do single gene testing. And the result, whatever that may be, whether it's positive or negative, is what it is. Sometimes we can order additional testing, many times we can't. But now what we do is we test for multiple genes simultaneously. So what we do is we order every single gene that has colon cancer in it, for example. And that allows us to really cast a much broader net. Because before, we had a patient who presents with a family history, and we would literally take a family tree and do this. This is what I saw for the first 15 years of my career. And now, with multiple gene testing, I can do this. I can see the whole picture. And that's why multi-gene panel testing has revolutionized the way we deliver genetic care. This is what the BRCA1 pathway looks like. You don't need to remember it, but you could see that it's right smack in the middle of everything. All those other little bubbles around it are other genes that can, if they don't work, interfere with that pathway. So they're all part of panels. For breast cancer, there's what we call genetic heterogeneity, because look at this. Look at all these genes that can cause breast cancer. Panel testing includes all of them. Well, what's the contribution? People say, oh yeah, okay, sure, but you know, how much of a percentage extra are you going to pick up? Well, it turns out a good amount of it, because Li-Fraumeni syndrome, 50% cancer by 40 years of age. P10-Cowden syndrome, there has an increase of almost 80% for breast cancer. STK11, there has all these other types of cancers. So I'm going through these really quickly, not to necessarily to skip through slides, but just to show you that there's a lot of information here. And these genes can predispose to many different types of conditions. So for example, just pick one. STK11, super rare. It's a gene that causes, traditionally, in my old world, what's called juvenile hematomatous polyps. It's so rare, it's like snow in June. But we are finding that in breast cancers. We're finding that in pancreatic cancers. We're finding that in colon cancers and in some lung cancers. So all of a sudden, this super rare gene that, I personally have never seen an abnormality in STK11. When we do broad testing, we're picking it up in all different types of cancers. Now, the question is it really causative? Just because it's there, is the there, right? Just because it's in a person with, say, ovarian cancer doesn't mean it caused it. And that's the real problem we're having now. This idea of connecting the genetic abnormality with a condition there. And that's what we're working through right now. There's lots of work being done in this area. Lots of different companies doing it, different multi-gene panels. It's a virtual maze of figuring out what you should order for a patient. There's over 127 genes that predispose to cancer that you can order. studies have looked at this over and over and say, well, okay, if somebody had BRCA testing, if you just look at a whole bunch of tumors, what's the likelihood of that you're going to get BRCA? Well, in this particular study, it's a famous study, the study that really began this whole idea of multi-gene panels, came from Mary Claire King. About 80% of people with these types of cancers that are here, you can see them here, had BRCA1 and 2. The rest, about 6%, had non-BRCA2. So breast ovarian cancer, pancreatic cancer, uterine cancer. They had other mutations in these other genes. And so the multi-gene panel saga began in 2011. Other studies, other groups have done their own research, and there's a whole bunch of probabilities and likelihoods, but really what you need to know is there's a gene called ATM, and ATM is a gene that typically is associated with neurological abnormalities, ataxia-telangiectasia mutata, and it's ataxia-telangiectasia is this abnormal gait. It's basically a neurological condition. It develops in childhood. It's recessive. I know I see Miriam. It's recessive, and so a researcher at UCLA in the 90s said, what's really interesting? All these children, who's studying ATM, all these children with ATM, if I look at their pedigrees, family tree, very important. If I look at their pedigrees, their moms, a lot of their moms had breast cancer. How does that work? Do you think it's a predisposition to breast cancer? And all of us in the genetics community are like, oh, no. Of course not. Of course not. It's impossible. One's a neurologic condition, one's a cancer predisposition. But it turns out that he was right. So ATM does increase the risk for breast cancer, and we now include it in those panels. PALB2 is that BRCA3 we've been searching for the last 20 years or so. It's another gene that's including on panels. CHEK2, that should say CHEK2, it's a misspell. CHEK2 is an old gene, it's been around for a long time. We never used to think it was important. Turns out that it is. It's included on the panels. BRIP1 was one that made a lot of news in our world about a year ago because it's a new high-risk ovarian cancer gene, part of those panels for women who have ovarian cancer and are BRCA negative. There's a whole bunch in there, and the important part of it is there's NCCN guidelines for all of them. You feel like that? Some days I feel like that. Too many genes, too much information. Cancer risk may not be very high. So you might be dealing with 12% risk for breast cancer or 12% risk for colon cancer. They may be unclear and they may change. You may go from, I think it's associated to, oops, it's actually not. Or like the case with ATM was, it may go from absolutely not a risk factor for breast cancer to, yeah, it actually causes breast cancer. But we're still learning. And that is definitely the wrong number for my pearl of wisdom. But sometimes in our quest for answers, we end up with more questions, and that has been the story of my career. And so I will stop here. There are some cases that we can go, if we don't have a lot of questions, we can go through at the end, and leave you with my this part, because there's a great slide. Leave you with that. Thank you very much for your attention.
Raluca, if you want to stay there, we do have several questions. In fact, over 20 questions have come in.
I talk fast.
If you have additional questions, please raise your card in the air and people will come by and pick those up. And I'm going to go ahead and start with some of the questions here that I have. This first one is asking, if parents are deceased and there's no genetic testing ever done on them, can you tell which parent was positive for the mutations, and does it matter?
That's a great question. So the answer is we can, and it does. And the way you can tell is by testing other relatives on either side of the family. So if the parents are deceased and the family history is on the father's side, as it is 50% of the time, you can test the aunt. You can test the aunt's kids. You can test other individuals on that side of the family. And if there is a mutation there, then the inference, and it is an inference, would be that that parent on that side of the family had the mutation as well. And yes, it matters because we need to know who else needs testing in the family. We need to know what side of the family we need to worry about. And when we can't tell, we test everybody, both sides. And sometimes you end up doing that, testing both sides in the quest of figuring out where it's coming from.
Okay. This next question is asking, how can two siblings with the same parents have two different DNA?
Well, they don't. They share 50%, two different parents? You mean, I'm not understanding the question. Or maybe I'm overthinking it.
I just have what's here on the card, but it just says, how can two siblings with the same parents have two different DNA?
Yes. So two siblings share 50% of the genetic information on average. Now, when you inherit your DNA from your parents, it isn't as pretty as I made it sound out to be. It's not this, you don't get one long string of pearls and then your sister gets the other long string of pearls 50% of the time. What ends up happening in the ovaries or the sperm is that the long string of pearls gets chopped up in pieces, really small pieces, and then the cells rearranges and I'll take this part and this part and this part. And that process is entirely random. It's called independent assortment, rule number 2 of Mendel's laws. And that independent assortment, on average, says that two siblings will have 50% identical DNA. Sometimes they'll have 75. Sometimes they'll have 40. Rarely they'll have only 20% identical DNA. But on average, it's 50%.
Okay. This question here says, I am BRCA1 and BRCA2 negative, and in 2015 I had another genetic test done. PALB2 showed up.
PALB2.
Is it something to worry about?
Yes, it is.
I'm going to ask why.
Okay. It's a court of law. You only asked, is it? Okay. Because PALB2 is BRCA3. PALB2 increases the risk for breast cancer. PALB2 increases, we think, and it's research still coming in, but it looks like it increases the risk for prostate cancer in men. It looks like it's a risk factor for ovarian cancer. But right now, the two cancers that are typical of PALB2 is breast and pancreatic. So if somebody has a true PALB2 mutation, they need to be followed according to the NCCN guidelines for PALB2, which includes annual MRI for the breast, includes probably some type of pancreatic screening, discussion of prophylactic mastectomy because the risk is significant enough in PALB2 to warrant that. So yes, it matters.
Okay, great. Thank you for answering that. This question's asking, how do you approach your patients that were adopted differently?
That's a great question. So I rarely refuse testing for anyone who wants it. So in the past, when we had adopted patients and we had these guidelines where you can only test these people, we would write letters of medical necessity to insurance companies and argue on our patient's behalf, saying this patient's adopted, there's no way they would know their family history, therefore I know she doesn't meet this guideline and this guideline and this guideline, but I think it's medically necessary she has genetic testing. Nowadays, that's much simpler because a lot of the criteria have been relaxed, and in fact, in NCCN, who should be tested guideline, which I didn't put up here, probably the only one I didn't put up here, is adopted. An adopted individual should have genetic testing, if they so desire.
Okay. This one's asking, if cancer occurs at an older age, can there still be hereditary mutations?
Absolutely. So that slide that I had of BRCA2, I don't know if for ovarian cancer, that showed the difference in BRCA2 and BRCA1. There was a red line and a blue line, and the blue line I think was BRCA2 and the red line was BRCA1, is a perfect example that shows that older ages of diagnoses can be hereditary in nature. And just as a caveat for that, because I think it's important, the reason we had these age cutoffs, because again, it's on average. There's always exceptions. And the more we learn about genetics, the more we learn that actually there's no rule. Everything goes. And so genetic testing should be something that anybody should be able to do or have if they want to.
This question sort of follows. It's a separate question, but it says, at what age should we say, so what?
I think that's an existential question. I'm up here because I believe in what I do. So that's never an option or never just something that I would do. I think genetic testing is powerful, and if for a particular individual at a particular age, they feel like they might not want to know, or there's nothing they would ever do it differently anyway, they may hold the key to information for other family members if they've had cancer. So doing the genetic test at that point in time is an act of selflessness.
Okay. This is a specific question that's asking, what are the genetics for sarcoma survivors?
Ooh. Well, it depends. Sarcoma is very complicated. There's lots of different types of sarcomas. There's soft tissue sarcoma, Ewing sarcoma, and only some of the sarcomas are hereditary. The majority of sarcomas are not inherited in nature. The ones that are hereditary are the ones that are seen typically in children.
Okay, this question is, we actually put this in two parts because they're asking about the environmental impacts on genes, what we eat, what we breathe, where we live. What are the mechanisms, for example, that damage the genes? And then the second question is asking, with proper diet and lifestyle, can you really change the DNA? So we sort of felt those were related.
Wow, how much time do you guys want to be here all night? Okay, so diet. No, I'm going to do two first. Okay, I'm going to answer number two first. So the dogma in genetics used to be that your genes are your genes and you're going to be what your genes say you're going to be. And that's just the way it is. And it turns out that's not true. It turns out that we found out that the DNA, and whether it's expressed or not, so whether the gene product is made or not, a lot of times is environmentally controlled. Now, before you guys get too excited, because I love hearing that too, most of the genes that are that have that switch for off and on with environmental control are not the genes we talked about here. These genes, BRCA1, BRCA2, the Lynch genes, the P53 genes, most of the genes we talked about here, when they don't work, the effect on that cell is so overwhelming that it negates anything else that might be happening. Having said that, even with all these genes, that only accounts for about 30% of all breast cancer. So sporadic breast cancer, right? That breast cancer at age 55 with not much of a family history, which is not hereditary, not inherited, probably can be amenable or could have been amenable to environment changes. Now, what are those environment changes? Well, we know that genes are turned off and on with this switch called methylation. There's this little CH3, one carbon and three hydrogen atoms, they all come to, it kind of sits like this, it kind of actually looks like this. It comes and it sits on the DNA and shuts down the gene, or gets off the DNA and the gene's active. And some of that is subject to a very complex methylation pathway, which is very hard to control, and we don't really have the ability to control it. But it is, in theory, controllable. There's one experiment that was done in Montreal maybe about 10, 15 years ago. It has nothing to do with cancer. It had to do with lab rats. And this researcher thought, well, I think behavior can be changed, and it's inherited, partly inherited, but it can be changed. So he took lab rats and divided them in two, took a litter, and then left the litter with the mom, and half the litter with the mom, and then took home with him the other half litter, which then he proceeded to raise on his own. And then he took them back to the lab and gave them a test. And the test for stress and ability to cope with stress and ability to be successful in the life of a rat is whether you can circumnavigate a maze to the cheese and how long it takes you to do that. And the more stressed out you are and the more unable you are to cope with your environment, and so not suited to survive, the longer your time is to get to the cheese. And I mention this because it's a classic experiment, what we're talking about, which is environmental changes to the genes. So when he took the rat pups that were with the mom, they outperformed the ones that he had taken home by orders of magnitude. And then it took him about 2 or 3 years to figure out why. So it turned out that the pups that were with the mom, for the first 3 to 4 weeks of their lives, were constantly licked by the mother. Constantly licked by the mother. Now, he didn't lick his rats that he took home and took good care of them. But he didn't. So it is, but it is, and it was it made, it's one of the most amazing experiments in genetics, because what it showed is that this licking, it's a behavior. This licking behavior did something to the genetic makeup of the rat. And how he found that out is he sacrificed the rats, took out their brains, took out the DNA from their brains, and then look at what genes are turned on, the rats that were licked versus what genes were turned on in the rats that were home with him. It was a very different pattern. So the licking turned genes on, turned genes inside the brains of the licked rats. That did not get turned on the brains of the non-licked rats. Mother, yes. See? Genetics is fun, guys. Come on. Thanks for staying till the end.
Okay, so a few more questions here. This one's asking, what is the role of telomeron in creating mutations?
Telomeres.
Oh, okay.
Yes.
Sorry.
Telomeres.
Read it.
Okay, so telomeres actually have to do with aging. So telomeres are these structures, and I wish I could easily go back to my slides, but do you remember when I had the picture of the chromosome, and I had, I showed you guys there was the DNA all packed in this sort of like X-shaped thing that's called a chromosome? Anyway, there was it's like an X-shaped structure. We have 23 pairs, total of 46, all of us in this room. Telomeres are at the ends of those chromosomes, and they regulate how the DNA is accessed, like how it's unwound and accessed versus kept sealed and unaccessed. So telomeres control to some degree aging. We really don't know how. And as far as I know, they're not a cancer risk, if that's the question. They don't play a role in cancer risk, but they control how fast a cell ages. So perhaps indirectly, they play a role in DNA fidelity, DNA transcription or DNA copying fidelity, right? That copying process, making it as accurate as possible.
And this might be related to that question above. It's on the same card. It says, what might be the effect of gene editing and replacing, quote unquote, bad genes? I don't know if that's related or a separate question.
There's no such thing as a bad gene. As an aside, there was a study that was done just randomly on hundreds and hundreds of individuals that showed we all carry about 4 to 5 lethal abnormalities that are recessive. So if we happen to find a mate that has that same lethal abnormality or one of those same 4 or 5 lethal abnormalities, then we can have an increased risk of having a child who either is never born or dies young. So there's no such thing as a bad gene, but the gene editing is a hot topic, and I want to come back and talk about that another time. But about a year and a half ago, this wonderful researcher, whose name escapes me, she's lovely, discovered by chance this enzyme called CRISPR-Cas9, We have very inventive names in genetics. I don't even know what it stands for. But it was revolutionary, and it was one of the biggest things that has happened in medicine in the last 100 years. And what CRISPR-Cas9 does, which we've never been able to do before, is with surgical precision, cut out pieces of the DNA and replace them with other pieces of DNA. We've never been able to do that. We were able to use retroviruses and these sort of vectors and other stuff to kind of nudge other DNA to replace, good DNA to replace the bad one, but it never stuck. And so CRISPR-Cas9, with surgical precision, takes out chunks of DNA, which anybody can design what chunk you want to take out, and puts a different chunk in. And it has unbelievable potential for curing human disease. It also has unbelievable potential for being misused. We in this country got together as a society and promised we would not use it to edit the sperm or the eggs, which would basically be creating designer humans. However, there are countries outside of the US that are currently doing that. So it's a very loaded question. I probably can talk about it for hours. But I think that answers it, yes?
Yeah. And I think we have 3 more questions here that we've narrowed down for time's sake. This one's asking, what is the percentage risk of getting a second cancer, breast cancer, when your grandmother and mother both had breast cancer and not be BRCA positive?
Okay.
So what is the percentage risk of getting a second cancer, breast, when your grandmother and mother both had breast cancer and not be BRCA positive?
I don't know. I don't know.
Could you work that up in your office?
No, I could not. The reason I don't know is because that is, we've not done that research. So if somebody is BRCA negative and has a family history of, has a personal history of breast cancer, has a family history of breast cancer, that's what it sounds like. Am I reading that correctly? What is the risk of a second breast cancer? Well, usually the risk of a second breast cancer is given based on the first diagnosis. So if the first diagnosis was under age 35, I think, then it's like a 2% for each year of life. If it's over age 40, and this is reaching way back, I think it's 0.5% per year. So you add that for every year of life. So if you've been diagnosed with breast cancer under the age of 35, then you add 2% per year of life. If it's over age 40 or 45, then it's 0.5% per year of life. But it doesn't have anything to do with the family history. Those two things haven't been connected. It goes by age of diagnosis of the first breast cancer.
Okay. This question here is asking, says, I see most results show insignificant. So based on this presentation, the relatives of the patient should be tested. The question is, if relatives are not tested, how does the patient manage at this point?
Okay, so ask me again from the beginning.
So the results show insignificant. So based on the presentation.
So insignificant, I'm going to presume just that means negative.
Insignificant test results is the way, I'm reading this. And I guess the question really is, so if the relatives are not tested, how should the patient manage at this point if nobody in the family is going to be getting tested?
But the patient herself has been tested and is negative, or himself.
It says insignificant. Yeah.
So if someone who has had genetic testing and is negative, and who has relatives who should be tested who refuse or are inaccessible to testing, that individual needs to manage their risk based on their family tree, the genetic biopsy. Take a thorough family tree. If the family tree says that grandma had breast cancer at 35, then she had breast cancer again in the other breast at 45, and then she had ovarian cancer at 56. And that patient is negative for all tests that we've been able to do, not just BRCA1 and BRCA2, then I would manage that patient as high risk. Because assuming that the information in the pedigree is correct. Remember, you got to make sure that it's correct. Abdominal cancer a lot of times stands for something else. Make sure the breast cancer was invasive and not DCIS. There's different risks with DCIS. But, I had a professor years ago who would say, phenotype trumps genotype. Phenotype is what the person looks like, right? So this is in the old genetics when we would do Down syndrome, and we would say, to see a child, I'm like, oh gosh, that doesn't look like Down syndrome. I don't know what that looks like. And by the way, I don't know what the test results are. We'd have a child who would say, oh, this looks like Down syndrome, and we'd do genetic testing, and lo and behold, it wasn't Down syndrome. You'd always say, phenotype, the way that child looks, trumps genotype. So ignore the test, go by what the phenotype is. In this case, go by the family history. Yeah?
Yeah, and then this will be our last question. Perhaps Raluca will be willing to stay a little after if there's some personal questions. But this one's asking, does 23andMe include any information regarding breast cancer?
Well, it depends on when you had it. Did you have it pre-FDA or post-FDA? No. DTC testing, which is direct-to-consumer testing, is another important topic, and I don't want to short shrift anyone here, but 23andMe is a valuable tool for many reasons. However, it is not a valuable tool for breast cancer genetic testing. I wouldn't use it as a way of changing management. So if I had a patient who came in with BRCA results from 23andMe, I would repeat them in a clinical lab just to confirm that there is a there. But yes, before the FDA got involved, they would give out breast cancer risk information.
Well, thank you so much for answering all those
Thank you guys. Thank you for your
questions.
attention.
Please,
Thank you for being so patient.